Let's go ahead and start the next session. Excited to kind of shift gears a little bit from, as we look from the payer model, to start to focus on a different area, in this case, tissue repair and regeneration. And got a great group of panel here together today to kind of look at where we've been in terms of the last year and certainly the progress in this novel tissue therapeutic space. So with that, why don't we just go around the table here and have each of you introduce yourself, the company, kind of a brief background on that, and then to help set the stage for this also, what stage of development are you currently at? So, Petter, maybe we'll start off with you.
Yes. Hello, everyone. It's a great pleasure to be here speaking to you. So I'm Petter, or Peter, as some of you may say. I'm from VERIGRAFT. I'm CEO of the company. We are a Sweden-based biotech company. I have a background as a scientist with a Ph.D. in cell biology. I worked 10 years for AstraZeneca. I then got interested in stem cells and cell therapies, and at that time AstraZeneca was not interested in that at all, so I left the company and started in the small industry. I worked there for almost 25 years now. The last 10 years in this company, VERIGRAFT. So we are in early clinical development. We have a first-in-man trial ongoing in Europe, and I'm sure I will have the opportunity to comment a little bit more on that today. Thank you.
Excellent, thanks. Don.
Great. So my name is Don Fink. It's a pleasure to be here. What a great venue. This is so nice, and it's great to be here in Rome celebrating with you this great event. So I'm an ex-FDAer, and that doesn't really carry as much terror as it did when I say I'm from the FDA. So I'm an ex-FDAer, but I now work in private consulting, and while I was at FDA, I was in what is, you know, as the Office of Therapeutic Products, Division of Cell and Gene Therapy. My expertise was CMC reviewer from front to back. And what I'd like to do, so obviously I don't have a product per se, but what I can tell you is there were some, while I was there for nearly 30 years at the agency, there are some prototypes that are going to sound pretty familiar when you think about what we're talking about today in regenerative medicine and creating these constructs. So if you go back a ways, to a couple that were cell therapy based, one was called Gintuit, and that's an allo product with keratinocytes, fibroblasts, and they're cultured on bovine collagen. And then you had Macy, and Macy was already a product in the market that was then brought into under regulation, and it's for knee repair, and that was for autologous chondrocytes. Again, in a membrane, this one was porcine collagen. And then you have Stratigraph, which was an allogeneic cultured keratinocytes, and that was for skin, murine collagen as a scaffold. And then there's also a few products that were regulated as devices that have been shifted over to the Center for Biologics. And they all have a common theme. There's Apligraph, Orcell, Dermagraph, Epicel. And all of these are cell-based products, either in conjunction with a base matrix, such as collagen or something of that nature, or as sheets, and these were all for skin repairs. I call them prototypes, first generation. I think that we're now outside that box and looking forward to creating more complex constructs as we move forward.
Thanks, Don. Tom.
I'm Tom Lowery, President and CTO of Satellite Bio. Prior to Satellite Bio, I'm an experienced executive that's led first-in-class technology development through the FDA, 7 products in the device space. Prior to coming to Satellite, I was one of the founding executives at Satellite in the summer of 2020, based on technology out of MIT and BU. At Satellite Bio, we're focused on severe liver disease, and we're pushing forward 2 technology breakthroughs. One is a sustainable and scalable cell source to treat patients with hepatocyte and liver disorders, and then also a tissue seed technology that stabilizes those hepatocytes, helps get them to engraft, and create a satellite liver in an ectopic location where we leave their diseased liver in place.
Great. And then I'm Jeff Ross, president of Miromatrix, where our mission is to bioengineer transplantable organs. And the way that we do that is we take an existing porcine organ, strip all the cells out of it, and then recellularize that with human cells. And we just announced earlier this year that we got our IND cleared for our bioengineered liver, first starting outside of the patient before moving forward with our products of fully transplantable kidneys and livers inside of patients. So you can start to see the progress in the field. I think just through the introductions here, you can see the broad swath as well when we think about tissue repair and tissue regeneration, whether that's strictly a cellular-based, or you can also see across the panel the combination of a scaffold, such as a biomaterial, in cells. And I think through this discussion today, we'll certainly get into a little bit of what foundation has been laid. I think, Don, you highlighted that a little bit of some of the predecessors in this space that have really started to tee up some of the advancements that we'll see inside of this space. So I think with that, let's just dive right in and maybe go around again and just highlight a little bit to kind of cue up this session and talk a little bit more about this. As we look at tissue repair and regeneration, you can see we all come at this a little bit from different aspects. But why don't you describe the progress that you see in the field? And maybe we'll go in reverse this time. Start with you, Tom.
Sure. I've been really excited to see the progress in the type 1 diabetes space in this field, where they're using beta cells or islets to treat patients that suffer from severe diabetes. And that's helped, I think, companies like ourselves, because it establishes clinical and regulatory precedent to how to manage patients that have receiving complex therapies in terms of are they going to receive immunosuppression, and also help educate the regulators in terms of how to deal with the non-clinical CMC and clinical packages. So that really helps pave the way for new novel cell types like hepatocytes.
So I think the really key piece for the regulatory perspective in connecting with all folks that are innovating in this space has been the sophistication and development of new tools for answering questions that were largely black box questions before. And this is really in the interface between materials. And I'm going to be speaking about here, what I'm really referring to is multi-complex, probably something, we'll call it a combination product, which has a scaffold of some type and a cellular product of some type brought together to form this tissue construct. There's always been questions like, what happens when they interface? What can we read about the cells after they've come on a dynamic surface and responded in some way? That does it alter their properties? And these have all been questions that for a long time we just had to be guessed at. But here now there's tools that you can actually look at very discreetly, all the way down to genomic levels, changes in cells based on interactions with different kinds of materials. And that gives the regulators, well, two things. One, a lot more questions they can ask you, but also it gives them a chance to have a more fuller understanding of what are those key issues that are going to need to be addressed as you go through product development. So that's kind of what I see as a really neat development in advance recently.
Better?
So, tissue engineering and tissue therapies are of course, a little bit of cell therapy as well. We always have cells there, but we also have something else, something where the cells are growing on or helping the cells to do a good function. We typically call that a scaffold. And that scaffold is something that is, of course, close to my heart, but also something that has advanced quite a lot last couple of years. So for some of you, scaffolds may be something simple, but scaffold can be a lot of different things, a variety of things spanning from human-donated scaffolds that we can use. It can be from animals, being xenogenic scaffolds. And today, there is something that is really rapidly advancing, and that is bioprinted scaffolds or printed scaffolds. So that is something that I see are really taking off now and are giving us, doing bioengineering, really good tools for create the products that we would like to take to the market.
Could it just quick follow on to that? I love this idea about bioprinting because I think it tends to describe the advancement of sophistication. And so what would be the most sophisticated sort of thing you could make with bioprinting? I mean, in terms of pore size, in terms of the cytoarchitecture, what is the most compelling piece about that?
First of all, one should maybe think about wording here. I'm a little sloppy myself. So when printing a scaffold, you should say 3D printing. Bioprinting is more the... So I think what I referred to was more the scaffold now, but bioprinting will also partly be the cells.
I think we keep losing.
Yeah, we'll have a time.
Move your mic a little bit
You're going to get a new mic. Sorry, I didn't mean to put you on the spot there. Go ahead.
Yeah. As you get remiked up there, Petter. I think as we look at the progress inside the space too, certainly I think there's been a lot of progress. We look at companies like Humacyte now, getting ready for a BLA. on more of a scaffold type approach, but still starting to look at tissue therapeutics and how we look at starting to address some of these things and bring function back or cure. As well as I think just the tremendous de-risking. I mean, Petter, you're in human clinical studies. We just got our IND cleared. I think what we're starting to do for the field and see across the field, and this goes to what you said a little bit, Don, is you're starting to see the de-risking taking place, where 3-4 years ago, you know, the comments were, this is really complicated. You're gonna have a biomaterial, you're gonna have cells, you're never gonna get through to human clinical studies. There's too much to do. And I think now we show that there is a path. And now with that path starting to be forged, I think that's gonna open up a lot more innovation on the backside, because we know that the regulatory agencies are open to this, they're willing to work together, and they're providing a path forward. So I think as we shift gears and think about that a little bit, let's talk about regulatory a little bit, because that often is the first hurdle as we look at this, and certainly important for the progress in the field. What has your experience been progressing, whether that's through the FDA or whether that's through notified bodies? Maybe we'll start with you, Petter, give the European look on this and what your experience has been on the regulatory side, both with your current product, but some of the products you're also starting to think about.
Yeah. So I'm the European guy here in the panel, so I will, and my experience is from Europe so far. So I would start to say that the regulators and the authorities, they have been very, very constructive and very easy to talk to, I should say. And I mean, the first thing was to define what our product really is. And the first product, I mean, is a classical bioengineered organ or a tissue, so it's scaffold and cells. And since we are dealing with cells and using cells in our manufacturing, it's clearly an ATMP, and thereby it has exactly the same regulatory status as a cell therapy. So that's very simple. Next was when we planned for the first-in-man trial, what the regulators said, there was one very logic thing, and a thing that I like. We were asked to have a staggered inclusion, and that is very logic, because we were allowed to transplant the first patient, then we followed this patient for a follow-up period, safety follow-up period for a little while, and then we got a clearance. An internal and an external advisory board said yes to go on, and then we were allowed to treat 2 more patients, follow-up period again, 4 more patients, and a follow-up period, and now we have a go for the rest of the trial. So that was very pragmatic and very useful, I think. So I think throughout this, they have seen that we are doing an autologous product, since we are personalizing our tissue engineered graft, and that was straightforward. So we haven't really faced any particular challenges, I would say.
And how many patients have you enrolled so far, and how big will your study be?
So it's a small phase 1/2 trial with 15 patients, and we have enrolled 60% of the patients. But now, after the last safety follow-up period, we expect the recruitment to go much quicker.
That's excellent. Don, maybe your perspective from an FDA standpoint, and maybe a little bit on the combination product and what that potentially means as well.
Well, yeah, I'll come back, I think, to the combination piece. So a combination product is simply a definitional term, which adds regulatory complexity in this context, because we're talking about things that are regulated by 2 different entities within the agency typically. Although I can say what I really wanted to say to that point is the reorganization of the Office of Therapeutic Products is actually to your advantage now in this space. They've been able to create their own tissue engineering branch. And remember, when I was back there, I was tasked to pretend I knew anything that would be contributory to a combination product that's going to be a tissue engineered product. So that took me out of my depth. But here you have folks that are often biomedical engineers, folks with biomaterials background experience, with engineering experience, that are now integrated into the review process from the beginning within the Office of Therapeutic Products. So I think that's a magical, actually, moment that we haven't recognized yet. But going forward with their experience, it's going to become very clear that when you have a group of folks that are focused in a specific area, they're going to have much more bandwidth of knowledge to be able to bring all of that experience to your product in a way that's going to be productive. So that regulatorily, I think, has been probably an advancement that we can't fully appreciate yet, but I project will be very important to the field. I did want to come back. Did you have, what kind of pre-conversations did you have? Did you have any scientific advice, or did you have that? Did somebody here have parallel scientific advice? Has anybody had that? Have you had that?
Yes, we have a little bit of that, but not much. It was relatively straightforward for us.
Yeah. I think parallel scientific advice, I love that concept. I didn't have much chance when I was at the FDA to be engaged in that exercise. I would encourage folks to really pursue that if you can, and it makes sense because it gives you a hands-on first look, both agencies or both regulatory bodies bringing the same kinds of looks and eyes and questions so that you know at the kind of at the beginning if you're intending to bring your product into the U.S. Or vice versa, if you want to go U.S. to the EU. I think parallel scientific advice can be really constructive, and I hope people will use that more frequently. I think it's a useful tool.
Tom, how about your experience on the regulatory side?
We're midway through the experiment of a parallel scientific advice. So we run the first arm of the experiment, which we just got pre-IND feedback from the FDA. And then in June is when we'll get feedback through the SNSA process in Europe. Exact same briefing book with some region-specific tweaks to it. Our drug product is cryopreserved, so that gives us a lot of flexibility in terms of what region we'll do our first-in-human trial in. I think to what Jeff said, our experience with the pre-IND feedback that we just got really supports the concept that the early days may be over now, and that there's a really sort of depth of expertise and understanding in the agency at the FDA. And then I think to your point, Don, from a staff perspective, we really had what I call sort of reciprocal exchange with them, where we submitted a very detailed briefing book across clinical, non-clinical, and CMC, over 150 pages. And we got a very detailed written response, almost 30 pages, that was all extremely reasonable, very well thought out, and excellent feedback that was right on target. We didn't have anything in there that were like, what, this doesn't make sense, they don't understand our product. And that really laid a roadmap for the data package that we have to assemble. I think one of the big riddles for these kinds of products is the non-clinical package, and you end up stitching together a mosaic of different animal models, and that's one of the key sort of sets of questions, other than, of course, your clinical study and CMC. But I think there's more precedent to look to on the CMC side for these kind of products. But on the non-clinical side, I think I'm seeing from the agency the ability to grasp the complexities of balancing that mosaic model and trying to help us deal with the limitations that sometimes we deal with when we try to demonstrate the product performance along all the attributes that we need to.
Yeah, maybe let's dive into that a little bit more, because I mean, as you do look at the briefing book, and certainly as you get submit your IND, the preclinical model is a huge component of it. And we're talking about therapeutics such as this, tissue therapeutics within this space. Often we're developing a human therapy that we need to demonstrate, whether it's efficacy or potency or robustness, in a preclinical model. So in many ways, we also have an immune challenge associated with that, unless we're doing a surrogate. So just interested in the panel's perception on this and how you've worked past that in terms of preclinical models. Maybe Tom, since you brought that up, maybe get your perspective a little bit on maybe whatever you can share on what you're doing or your guys' approach on that front.
Absolutely. I think that if you haven't been in the thick of tissue engineering, you may not appreciate how complex the preclinical models have to be. You have to show durability, and that your product is durable and integrates into the tissue like it should. You have to show safety of your route of administration and do that at scale. Those two things tend to lead you to non-human primate and pig models. And then you have to show efficacy, so you need a disease model, and sometimes you need multiple disease models depending on the functionality of your product. You have to show safety and tox, and those end up all being different categories of models. My experience has been if you try to ask too much from one model, then you're destined to have a lot of serial failures. The other strategy that we've employed successfully is having parallel paths in terms of our model development. So doing all the diligence you can in terms of all the possible models, particularly for efficacy, but also for these other models, and then trying to do the development work, knowing that you're actually going to need to present negative data for each model to support the models that you do select. Because conceptually, without that data, it's easy for folks to say, hey, why didn't you do the model this way, or why didn't you use this type of animal? And so collecting that full data package, both that support your selected models and show that the other potential models don't work, is an important process. This is going to be a rate-limiting step for any company in this space unless you start early, do parallel processes there, and then just embrace the rigor. And I think that on the other side, on the regulators, I think there is flexibility as long as you have a data-driven approach.
Yeah, I mean, I would certainly highlight that. You know, our own experience in that is do parallel paths on multiple models because each has their limitation. And I think some of the progress in this space certainly is just seeing the FDA lean into this. And that is if you can show data and the limitation of a model, they're much more open to the next model or piecing together multiple models to really show that total package. So I think it's a misconception that you need one model to show all this. Many cases, you might have 2 or 3 different models looking at this at different areas and different aspects to be able to achieve that. Petter, what's your experience in this?
Yes. So luckily, the tissue engineered product we are having in the clinic now is an example of a product where the tissue itself is giving the function. So we haven't discussed a lot about potency with the regulators. Instead, they have focused almost 100% on safety. And that is, of course, very important when it comes to a whole organ when you introduce that. And they focused on 4 areas. So they asked us to show both preclinically and now in the clinic, one, rejection. And that is the whole thing with our technology, that we want to avoid rejection. Second is infection, very important for introducing a new tissue like this. Third one is thrombosis, because our product is in the vasculature, so thrombosis is very important. And the fourth one, and most logic maybe, is mechanical failure. So all kinds of failures like bursting or leaking or whatever. That is something that have asked us to really carefully assess. First, of course, carefully preclinical, but now also in the first-in-man trial.
And I think these are really 2 really great examples of the type of meeting that would be a benefit if you're a new innovator, which is the Interact meeting, because the Interact meeting is really intended to assist early development in the preclinical space and the crucial models, particularly around, it's so complicated with a matrix, but to get exposure to agency thinking about how you can establish the bona fides from a preclinical perspective. It will help guide you when you go forward to the pre-IND. So it's a sweet spot. We've been relatively successful at Dark Horse at getting folks into an Interact meeting. And you have to realize it's only been about 30% success rate for that. So, and there's a lot of reasons for that. But I think these are both classic examples of the types of questions. And the other thing that you mentioned, Tom, that's important, I think, is to understand the agency will always say this: remember, you need to use the product that you actually intend to give to the patient. Well, there's not a lot of animals you can put something of scale into to make that actually happen. So you have to have a convincing discussion or conveyance about prototype, scaled down versions, the extent to which those are going to replicate what you might expect in a larger human context. And that's a challenge, particularly if you're still innovating that product to try to get it to be its optimal version that you intend to go forward in the clinic. And because if you change after what you've done all your preclinical work on and say, now we're going to use this version in the clinic, you're going to have to go back and show me that that's okay.
Yeah. Or do a comparability study and demonstrate that, which can work, but is challenging as well. Hey, you had made a comment previously in one of our conversations, Don, and it'd be great if you could expand on this a little bit, and that is around the notion of, sometimes the preclinical model is not going to take you all the way, and talking about what you can do in that phase 1 clinical study and how you queue that up sometimes to answer some of the questions that you can't do in the preclinical.
Wow, you remember something I have nothing. So, what you can do, I think, in the early trial space, and I think we heard a little bit about, is how you design the patient enrollment and how you look at the key questions that might be of concern toxicologically, and to get buy-in about how we can go in and look and read out and take care to sort of to reinforce the events that we think might happen that we can't actually test in the animal model. Because the animal models, I think what I'm remembering now is the animal models are often, they fail to really signal the types of events that could occur in the human condition because of all of the things you've just heard about not being similar, reacting to the materials, of being immunosuppressed in order to tolerate human, or having been genetically engineered. So you have what you have as baseline information, but then you construct a conservative approach to look carefully and fully and robustly at outcomes of safety as you progress, and then stepwise progress forward from that. So you can work with trial design and clinical review to make sure that something like that can happen.
Excellent. We do have a couple minutes left before we wrap up. I got a couple more questions here, but any questions from the audience?
Cube, I love that, the cube.
Always hard to see out there, but looks like there are none right now. Let's maybe spend a couple minutes just on manufacturing and potential the progress in manufacturing, and probably hit Petter and Tom on this a little bit. Maybe describe a little bit of your manufacturing. Is that in-house, or did you go CMO with it?
So what was the question there?
Manufacturing your product.
Yeah, I mean, in general, that is something extremely important for all of us. It's more complex than any other therapy that I can think of, at least. So you have to use partners. You mentioned CMOs that are really having some experience from this, because it's not like only manufacturers cell therapy. It's much more complex than that. Depends on the product, of course, but we have been lucky to find partners in Europe that are skilled in the art and that can manufacture for us. So all good now.
That's excellent. Tom?
Yeah, we've chosen to keep all our manufacturing in-house through our phase 1/2 trial, and that's largely driven by, A, we have access to a great talent pool in the Boston area, and 2, we have to figure out this manufacturing process very early and then establish that. So early on in our R&D, we already implemented a batch record approach and COTs. We have a small manufacturing team that has experience from even commercial stage cell therapies that we brought in to help mature the internal processes and really implement quality by design principles from the get-go, and then make enough batches frequently enough to be able to refine that process. And then at the same time, very early on, we dove into COGS minimization to be able to help reduce that development cost and our per batch cost. And over the last, as we've sort of caught our wind on that, over the last 12 months, we were able to really have a significant decrease in that COGS on a per batch basis. So we're a small team, about 35 folks, and we have, interesting enough, on our liver isolations, when we go live, we have 2 teams of 5 folks that each spend a 4-hour shift that can come anytime during the day, and people are thrilled to be a part of that isolation team. We're hoping the enthusiasm lasts, but it's been a really big help to us because of the inherent complexity of the technology and having to figure it out as we move forward.
We had a similar approach, early on, really looked at what's the ability to do that in-house versus outsource. And I think the uniqueness of this field really pushed at least the early developers in this phase to really have to do the manufacturing process in-house because it's so complex. I shouldn't say complex, it's just different. Unique. There's not a lot of tools and ability to outsource that yet today. So I guess with 2 minutes left, it'd be good to just get your final thoughts on, what excites you about the future for tissue repair and regeneration? Maybe we'll start here, Petter, go around there, and I'll close it out.
Yeah, so I think the timing of your question, Jeff, is fantastic for me because the timing of this conference and this panel is giving the answer to that, because 2 days ago, I was asked the same question by a journalist. He asked me, what is the most fantastic moment over these 10 years? And that was, of course, I should say, the day when we transplanted the first patient. So that was absolutely fantastic when you saw that you could, with the tissue therapy, turn a serious disease into a healthy person again. So that is absolutely fantastic. And I don't think there is anything like this area out there. We cannot treat some diseases with cell therapies or gene therapies or small molecules. That can only be done with tissue therapies. And tissue therapies are, of course, the most sexy you can ever do in life.
Go ahead.
Are you pointing at me for being
I'm
sexy
pointing
or something?
at you. You're a product man.
I'm a product man, yes. So what I've been really excited about is when you look at what's going on with CAR-T and the standardization and all the different disease indications that's tackling, is we see these more complex tissue engineering approaches benefit from that standardization and benefit from products advancing deeper into the product life cycle and through clinical trials. The number of patients that we're going to be able to treat just explodes. A hepatocyte has 500 different functions, and there's a huge unmet need there. I think we see it in type 1 diabetes, and a lot of other areas for tissue therapeutics. And so as this standardization occurs, I think we're going to see a really exciting explosion of patient impact.
I think just to build on that a second, I mean, we saw earlier today talking about autoimmune. So, you know, we go from how the field has evolved in terms of CAR-T to started to open it up. Always seen tissue engineering as kind of that next phase. How do you take the lessons learned in that? And to your point, Tom, and to your point as well, Petter, is, you know, we start talking about what tissue therapeutics can do, and you mentioned some of those, but we start thinking about renal failure. You got 500,000 patients on dialysis. You start looking at these much broader patient populations and where this stuff can go. That really excites me about the future. Now that we're starting to see these therapies get through their INDs, and we're starting to see phase 1, to your point, I mean, that's just a glorious time when you start treating patients, and we can start to show the potential of this now to affect and save lives. So appreciate everyone's time on the panel, and please help me thank them.
Thank you.
Thank you.